In this video tutorial, I am going to discuss about Hexose Monophosphate Pathway. It is often abbreviated as a HMP pathway. Now, HMP pathway is the alternative oxidation pathway for the glucose. Now, we know that the glucose, the main oxidation pathway is not the HMP pathway, rather mainly glucose undergoes oxidation by the glycolysis, which is followed by TCA cycle. And by this main oxidation pathway, glucose gets completely oxidized and broken down to the carbon dioxide. And during this oxidation, energy is released in the form of ATP. This HMP pathway is the alternative oxidation pathway for the glucose. So here what happens? Glucose undergoes oxidation by this HMP pathway. And here also, glucose is completely oxidized and broken down to carbon dioxide. The difference with this main pathway is that here in this HMP pathway, energy is not released. Rather, two important molecules are synthesized. One is NADPH and second one is ribose 5-phosphate. Now, as this ribose 5-phosphate is one of the very important molecule, so to give recognition to this molecule, this pathway, it is also called as a pentose phosphate pathway. Pentose phosphate pathway. And if you notice one thing, that this is the alternative oxidation pathway for the glucose. So what happens? This HMP pathway, it takes away this glucose away from this main oxidation pathway. So sometime instead of pathway, it is also called as a shunt. So Hexose monophosphate pathway, instead of that we can say Hexose monophosphate shunt. Now if we talk about location of this pathway, it occurs in the cytosol fraction of the cell. And if we talk about at the tissue level, this pathway occurs in all the tissues. However, in some tissue, HMP pathway is more active. What are these tissue? So to understand about this tissue, first we need to understand what are the functions or which tissues require more of the NADPH and more of the ribose 5-phosphate. See, NADPH in our body, it has two main functions. The first function is in the reductive biosynthesis. In our body, there are mainly two molecules which are synthesized by this reductive biosynthesis. One is fatty acid synthesis and second one is the cholesterol synthesis. And once cholesterol synthesis occurs, it also gets converted to the steroid hormones. So, steroid hormone synthesis. Fatty acid synthesis mainly occurs at three places. The first one is liver, second one is adipose tissue, and third one is lactating mammary gland. Whereas this cholesterol biosynthesis, which followed by steroid hormone synthesis, it occurs in mainly three tissues. The first one is liver, which is common to here also, then adrenal gland, and third one is the gonads. Gonads means testis in the male and ovaries in the female. The second important function of this NADPH is to counter free radicals or oxidants. So, to counter free radicals or oxidants. And this, to counter the free radical and oxidant, it is required by all the tissue. However, it is at most important at three places. The first one is RBCs, that is red blood cells or erythrocytes. Second one is the lens and third one is the cornea. If we talk about ribose 5-phosphate, it is one very important function that this ribose 5-phosphate, it acts as a structural component of DNA and RNA. Now we know that DNA and RNA, this genetic material, is required more by the tissue which is undergoing rapid cell division. So of course, this ribose 5-phosphate is required more by the rapidly dividing cell. And in our body, there are some cells which are rapidly dividing. What are these tissues or cells? The first one is the bone marrow. Second one is the skin. Third one is the intestine mucosa. These are all normal tissues which are undergoing rapid cell division, and one pathological tissue, that is the tumor. In the tumor, there is uncontrolled and rapid cell division. So, tumor pathologically will require more of the ribose 5-phosphate. So, as we know now that this tissue, and these all tissue, they require these products of hexose monophosphate pathway more often than the other tissue. So, this is the list of tissue which has more active HMP pathway. Right. Now, let us look at the reactions of HMP pathway. In the first step of HMP pathway, glucose gets converted to glucose 6-phosphate. This reaction is catalyzed by the hexokinase or glucokinase enzyme. This phosphate is being donated by the ATP, and ATP gets converted to ADP. Earlier in the video of glycolysis, we had discussed this reaction in the full detail. The entire purpose of this reaction in the HMP pathway is to trap glucose inside the cell. How? See, glucose is freely permeable across the cell membrane, whereas glucose 6-phosphate is not permeable across the cell membrane, and these enzymes, they are located inside the cell. So what happens? Glucose transported across the cell membrane comes inside the cell, where inside the cell this enzyme convert them into glucose 6-phosphate. Now, as glucose 6-phosphate, it cannot be transported across the cell membrane, as well as this reaction is irreversible. So now this glucose 6-phosphate is essentially trapped inside the cell. Now here there is no any transfer of carbon. So glucose itself is a 6-carbon compound, and so glucose 6-phosphate is also a 6-carbon compound. In the next important reaction of HMP pathway, glucose 6-phosphate undergoes oxidation and gets converted to 6-phosphogluconolactone. And this compound is also a 6-carbon compound. So here glucose 6-phosphate is undergoing oxidation, and it is coupled with the reduction of NADP+. NADP+ gets reduced to NADPH plus H+. Now this important reaction is catalyzed by one of the very important enzyme, that is glucose 6-phosphate dehydrogenase. And this enzyme is often abbreviated as a G6PD enzyme. Remember, this particular reaction is the rate limiting as well as a regulatory step of hexose monophosphate pathway. And this enzyme, it requires magnesium ions as a cofactor. Now in the next reaction, this 6-phosphogluconolactone, it undergoes hydration, that means water molecule is being added, and it gets converted to 6-phosphogluconate. This particular reaction is catalyzed by the gluconolactone hydrolase. Gluconolactone hydrolase. As you can see, there is no any transfer of carbon, so this is also a 6-carbon compound. Now in the next reaction, this 6-phosphogluconate further undergoes oxidation as well as decarboxylation. Now decarboxylation, that means carbon dioxide molecule is removed, and it is also undergoing oxidation. So here also oxidation is coupled with the reduction of this NADP+, and NADP+ gets reduced to NADPH plus H+, and simultaneously this 6-phosphogluconate is oxidized. It is oxidized to ribulose 5-phosphate. As you can see, one molecule of carbon is lost in the form of carbon dioxide. So ribulose 5-phosphate is the 5-carbon compound. Now ribulose 5-phosphate is one of the very important molecule. Why? The reason is that the ribulose 5-phosphate has a role in the regulation of glycolysis as well as gluconeogenesis. So glycolysis and gluconeogenesis. So basically this ribulose 5-phosphate is coordinating with the glycolysis as well as gluconeogenesis. Now in the cytosol, there are two different enzymes. One is epimerase and second one is the ketoisomerase. And for both of this enzyme, ribulose 5-phosphate is the common substrate. So if ribulose 5-phosphate is acted upon by this epimerase enzyme, then ribulose 5-phosphate is converted to its epimer, that is xylulose 5-phosphate. And again, this is also a 5-carbon compound. If ketoisomerase acts on ribulose 5-phosphate, then this ribulose 5-phosphate undergoes aldoketoisomerization to give rise to ribose 5-phosphate. This ribose 5-phosphate is also a 5-carbon compound. Remember, both of this reaction, they are reversible reaction. Earlier, all the reactions of this hexose monophosphate pathway, all of that reactions are irreversible. So before this ribulose 5-phosphate, all the reactions are irreversible, whereas after ribulose 5-phosphate, we will have all the reaction as a reversible reaction. Now you might be wondering that how one molecule of ribulose 5-phosphate can give rise to xylulose 5-phosphate and ribose 5-phosphate. Remember this very carefully, that one or single molecule of ribulose 5-phosphate can be converted to xylulose 5-phosphate or ribose 5-phosphate, not both. So to give rise to both of this compound, what happens? Multiple molecules of glucose undergoes HMP pathway. So multiple molecules of ribulose 5-phosphate will be generated. Out of these multiple molecules of ribulose 5-phosphate, some of them gets converted to xylulose 5-phosphate and some of them gets converted to ribose 5-phosphate. Right? That thing we have to keep in mind. Now in the next step, what happens? This xylulose 5-phosphate and ribose 5-phosphate both react with each other in the presence of enzyme transketolase. This transketolase is also very important enzyme because transketolase requires thiamine pyrophosphate as a coenzyme, and thiamine pyrophosphate is nothing but the active form of vitamin thiamine, which is also called as a vitamin B1. So what happens? Clinically, we can use this phenomena. Suppose in some patient you want to assess the nutritional status of vitamin B1. So at that time, rather than measuring directly vitamin B1, what we can do? We can measure the activity of transketolase. Remember, transketolase activity is dependent on thiamine pyrophosphate. So if this vitamin B1 is deficient, then transketolase activity will be decreased. So by that we can measure or assess the nutritional status of vitamin B1. And as we know that HMP pathway occurs in the cytosol and this transketolase is also present in the cytosol. So we have to measure this transketolase activity in the cytosol. And easily extractable cells are the red blood cell. We can just collect the blood and we can very easily get the red blood cells from the patient. So inside the red blood cell, we measure this transketolase activity, right? So that is the clinical utility of this enzyme. Now let us see what this transketolase enzyme will do to xylulose 5-phosphate and ribose 5-phosphate. What it will do, it will take up 2 carbon from this xylulose 5-phosphate and transfer it to the ribose 5-phosphate. So what will happen? This xylulose 5-phosphate, it will lose its 2 carbon. So now it will be 3 carbon compound which is called as a glyceraldehyde 3-phosphate. And this ribose 5-phosphate, it gets 2 more carbon. So 5 plus 2, that makes total 7 carbon. And here this 7 carbon compound is pseudoheptulose 7-phosphate. Remember, this entire reaction is the reversible in nature. In the next step, what happens? This pseudoheptulose 7-phosphate reacts with this glyceraldehyde 3-phosphate. And this reaction is catalyzed by the transaldolase enzyme. Remember, this transaldolase, it does not require thiamine pyrophosphate. Only transketolase enzyme, it will require thiamine pyrophosphate. What this transaldolase will do, it takes up a 3 carbon from this pseudoheptulose and transfer it to this glyceraldehyde 3-phosphate. So what happens? This glyceraldehyde 3-phosphate, its own 3 carbon plus 3 carbon derived from this pseudoheptulose. So it becomes a 6 carbon compound, that is fructose 6-phosphate. And fructose 6-phosphate can undergo gluconeogenesis to give rise to glucose. Here what happens to pseudoheptulose 7-phosphate? It is losing its 3 carbon. So 7 carbon minus 3 carbon, that makes total 4 carbon. And this 4 carbon compound is the erythrose 4-phosphate. Now this erythrose 4-phosphate and xylulose 5-phosphate both can react with each other by the action of the same enzyme, that is transketolase, and here also thymine pyrophosphate is required. Here what this enzyme will do, it will take up a 2 carbon from the xylulose 5-phosphate and transfer it to this erythrose 4-phosphate. So what will happen? This xylulose 5-phosphate is losing its 2 carbon. So 5 carbon minus 2 carbon, that makes up a 3 carbon compound, and here the name of 3 carbon compound is glyceraldehyde 3-phosphate. And this erythrose 4-phosphate is getting 2 more carbon. So 4 plus 2 carbon, that is 6 carbon compound, and this 6 carbon compound is the fructose 6-phosphate. And this fructose 6-phosphate, it can undergo gluconeogenesis to give rise to glucose. What happens to this glyceraldehyde 3-phosphate? See, glyceraldehyde 3-phosphate is the 3 carbon compound, and glyceraldehyde 3-phosphate, it can undergo gluconeogenesis to give rise to glucose. But what happens? For this gluconeogenesis, 2 molecules of glyceraldehyde 3-phosphates are required, because single molecule of glyceraldehyde 3-phosphate cannot give rise to 1 full molecule of glucose. So here to generate this 1 molecule of glucose, we need 2 molecules of this glyceraldehyde 3-phosphate. So now in this reaction, we need to generate 2 molecules of glyceraldehyde 3-phosphate, right? So this reaction should occur 2 times. So if this reaction occurs 2 times, then we can get 2 molecules of fructose 6-phosphate, and so we can get 2 molecules of glucose over here. So if this occurs 2 times, their substrate will also be required for the 2 times. So 2 molecules of this erythrose 4-phosphate required and 2 molecules of this xylulose 5-phosphate required. Now here we need 2 molecules of erythrose 4-phosphate. So of course this reaction should occur 2 times. So if this reaction occurs 2 times, we can get 2 molecules of fructose 6-phosphate and 2 molecules of glucose. And here substrate will be required doubled, right? So here 2 molecules of pseudoheptulose 7-phosphate is required and 2 molecules of glyceraldehyde 3-phosphate is required. Now for generation of this both product 2 times, this reaction should occur 2 times. So what happens? We need 2 molecules of ribose 5-phosphate and here 2 more molecules of xylulose 5-phosphate is required. Remember, these 2 more molecules were required at this step. So total 4 molecules of xylulose 5-phosphate is required and 2 molecules of ribose 5-phosphate is required. So to synthesize 4 molecules of this reaction should occur 4 times, and to synthesize 2 molecules of this ribose 5-phosphate, this reaction should occur 2 times. So 4 reactions this side and 2 reactions this side, we need 6 molecules of ribulose 5-phosphate. For this 6 molecules of ribulose 5-phosphate, we need 6 molecules of 6-phosphogluconate, we need 6 molecules of this 6-phosphogluconolectone. So we need 6 molecules of glucose 6-phosphate and 6 molecules of glucose. So here what happens? 6 molecules of glucose 6-phosphate undergoes oxidation to give rise to 6 molecules of this compound. So here 6 molecules of NADP plus gets converted to 6 molecules of NADPH plus H plus, and in this step this 6 carbon dioxide gets released, and here also 6 molecules of NADP plus undergoes reduction to 6 molecules of NADP plus. So the ultimately, after balancing this hexose monophosphate pathway, what we are getting? We are starting with the 6 molecule of glucose, and then we are generating some more glucose molecule. As you can see, 2 glucose molecule over here, 1 glucose molecule over here, and 2 molecules of glucose over here. So 2 plus 1 plus 2, that makes total 5 glucose. So what happens? Out of this 6 glucose molecule, we are already generating 5 glucose molecule. But along with that, what is happening? 6 molecule of NADP plus over here and 6 molecule of NADP plus over here, they are getting reduced. So we are all reducing 12 molecule of NADP plus to 12 molecule of NADPH plus H plus. Along with that, we are releasing 6 carbon dioxide molecule. So here 6 carbon dioxide molecules are getting released, and here remember this H2O, it will be also required of the 6 molecule. So this here 6 molecule of water molecules are required. So this particular reaction is the oversimplified version of the hexose monophosphate pathway. Now if you had noticed that there are 2 phases in the hexose monophosphate pathway. See, till ribulose 5-phosphate, what is happening? All the steps are irreversible. And along with this irreversible reaction, they are all doing oxidation. Oxidation is occurring. Here oxidation is occurring and here oxidation is occurring. After ribulose 5-phosphate, there is no any oxidation. So because of that reason, this particular phase of hexose monophosphate pathway, it is called as an oxidative phase. And this oxidative phase is mainly concerned with the generation of NADPH. Whereas after ribulose 5-phosphate, what is happening? All the reactions are reversible and there is no any oxidation. So this is called as a non-oxidative phase of hexose monophosphate pathway. One more thing, that here as you can see, ribose 5-phosphate is one of the important thing which is synthesized in the HMP pathway. So non-oxidative phase is mainly concerned with the synthesis of ribose 5-phosphate. So this thing we have to keep in mind. Now how this hexose monophosphate is regulated? So as I told you earlier that the main regulatory as well as rate limiting enzyme is this glucose 6-phosphate dehydrogenase. So this glucose 6-phosphate dehydrogenase is actually under tight regulation. This NADP plus, it acts as a positive allosteric activator of glucose 6-phosphate dehydrogenase, whereas this NADPH is the negative allosteric inhibitor of this enzyme. And insulin, it also affect the activity of glucose 6-phosphate dehydrogenase. What insulin does? Insulin causes induction. Insulin causes induction of this G6PD enzyme. So it upregulates the activity of glucose 6-phosphate dehydrogenase. So that's all about the hexose monophosphate pathway. In the next video tutorial, I will discuss about the significance of HMP pathway and the diseases or applied aspect of this pathway. If you have any query or confusion, please write it down in the comment section below. Thank you.